Welcome back to our session, which is guiding you for paper 2. We'll be looking at questions, most likely examinable questions, which are going to come in your paper 2, both in prelims and final exams. Don't forget that in most cases, we don't set new questions, but we use the questions which are prevailing, and then we modify them. So I'll be showing you how to answer these questions, and I'll show you different questions. Some of them I'll be just speaking about them, and then how do you answer those questions. So keep on so that you don't miss out anything, and don't forget to subscribe, like, and share to others so that you can also benefit. M.Saidi as usual. Let's start with the question 3. The diagram represents the portion of nucleic acids. Before I start this, don't forget that we have specific topics which we are supposed to look at in paper 2. The first topic is DNA, code of life. The second topic is meiosis. The third topic is genetics. The fourth topic is evolution, but both evolution in natural selection and then human evolution. So it means that these are the topics which are being set in paper 2. So when you are reading, you must focus on these 4 topics or 5 topics. So we're going to look at all questions, and then the most likely examinable questions or questions you're most likely to find in your paper. If they bring a diagram like this, you have to find out which diagram is representing. So this diagram represents DNA. Why DNA? Because of this, the weak hydrogen bonds. And then it's two-stranded. What are some of the questions which are most likely to be asked about the structure of DNA? Apart from labeling the structure of DNA, they can ask you to describe the structure. Apart from labeling it, describe the structure of DNA, and they put there 6 marks, whereby you have to tell us DNA is made up of nucleotides. Each nucleotide is made up of a phosphate, a nitrogenous base, a phosphate, and a nitrogenous base, a sugar, and a phosphate. It forms 2 strands. Each strand forms 2 strands. Each strand is joined together. So you describe the characteristics of DNA. Basically, what DNA has. It forms 2 strands which twist around to form a double helix. So you can get the 6 marks from there. So in this case, let's label it and then find out what is a V. What is V? V is the nucleotide, because a nucleotide is a nitrogenous base, a sugar, and a phosphate. So this is a sugar, and then nitrogenous base, and then a phosphate. So this circle or box is showing you a nucleotide. Then Z is weak hydrogen bond. Use the word weak. Weak. We want to see the word weak. Then hydrogen bonds. Weak hydrogen bond. And then this is a nitrogenous base. Nitrogenous base. And then also this is a phosphate. Sorry, a nitrogenous a sugar molecule, which is, in this case, it is a ribose, deoxyribose sugar. And then Y is a phosphate group, as we saw it here. So now, since you know that this is a structure and you know how to label it, now let's talk about name the nucleic acid which is being represented. So we have seen it that this is the and A. Then they're saying name 2 places in animal, say where you can find this DNA. Where can you find this DNA in animal? You can find in animal in 2 places. That is in the nucleus, and then in mitochondrion. If it's one nuclei, if it's one mitochondrion, if there are many mitochondria, if there are many nuclei, if it's one, it's called nucleus. So it should not confuse you. They are saying name portion V. They are looking for portion V. What is portion V? We say that these are the building blocks. This is the building block of DNA. So it is the nucleotide. Where is Y? Molecule Y is a phosphate. We have seen it that it is a phosphate group. Then they are saying that bond Z. What is bond Z? Bond Z is weak hydrogen bonds. So I think we have seen the answer here. Weak hydrogen bond, we have seen here, phosphate group, nitrogenous base, we have seen it, and then portion W, which is the nucleic acid. Sorry, which is the nucleotide. What is the natural shape? When they say natural shape, many students, they say helical, double helical. No, the only answer we accept in exam is double helix. Nothing else. You write something different from that, we won't give you a tick. Name the process during which this molecule makes exact copy of itself. How does DNA make exact copy of itself? How does it replicate? So the answer is DNA replication. So that is the process by which DNA makes exact copy of itself. Why does it occur? Some questions can come here. Why does DNA replication occurs? It occurs to increase or to double the chromosome number, Where does it occur? When you talk about where does it occur, then you talk about the nucleus. And then when does it occur? It occurs during interphase. Yes. And then why does it occur? Why does it occur? We say that to double the chromosome number. To double the chromosome number. And then lastly, how does it occur? That is the process of DNA replication. If they bring a question like this, how do you answer? This is a DNA molecule. So if you look at it here, you see it is a DNA molecule. And why is it DNA molecule? Because of the thymine, which is indicated in the nitrogenous base. And then also the weak hydrogen bonds, 2 strands, so it identifies it to be a DNA molecule. And it cannot be RNA, because even here they say that this is what? This is a DNA replication. Yeah, so question them. Molecule W, where is molecule W? We have seen it, molecule W is here. It is a nucleotide, correct. Molecule U, where is molecule U? The whole molecule is DNA, correct. Then they are saying that Z is weak hydrogen bonds, correct. And then what is V? How do we know the nitrogenous base V? Because nitrogenous base is compatible to this or complementary to this, therefore, and this is given it here, which is thymine, therefore V is equal to A, which is adenine. And then they are saying that name the part of molecule W is W, X, and Y. X is the phosphate group, correct, and then Y is the oxyribose sugar. Why don't I say sugar? you have to be specific because this is DNA. So it is deoxyribose sugar. Where in the cell does this process take place? Where do we find? We have answered this question that this process occurs in the nucleus. In the nucleus. And then they're saying that name the phase of cell cycle where replication takes place. We have already answered it, and then we say that interphase. So at least when they bring such questions, you must be able to answer accordingly. So first try to answer what is being labeled. The moment you know what the picture depicts or what a picture represents, at least you are able to bring your mind and then you concentrate in that area. I'm not going to spend too much time here, only maybe if I see a very difficult question there, then that's when I can try to answer related to this, because I wanted to go to transcription and the translation. But molecule C is phosphate, and then this is a nitrogenous base, and then this is phosphate, sugar, deoxyribose sugar, and then nitrogenous base. It forms what you call a nucleotide. And then weak hydrogen bonds, and then this is a sugar. This is a complementary base. So this is a complementary base. And then here, yes, they say this weak hydrogen bond. And then you have this. How do you call that? That is not a strand. We call it a backbone. And then the whole strand must include with the nitrogenous base and this. So the difference between a whole strand and a backbone is the backbone does not involve the nitrogenous bases. If you are asked what is this bond, this is a phosphate sugar bond, but it is a covalent bond. If you refer it to physical science. Okay. Two scientists who discovered, James Watson and Francis Crick, name one organelle where DNA is found in the nucleus and the mitochondria. Describe how mutation of DNA may change the structure of the protein. The moment DNA molecule changes, is going to change the sequence of the bases on messenger. And the messenger RNA is going to change the sequence of the amino acids in which they are being brought to form a protein. So if a different protein is formed, if the amino acid leads to a different sequence, then a different protein is going to be changed and or is going to be formed. Then amino acid leads to the same sequence as previous one. Then the protein is not going to, a new protein is not going to be formed. Tablet 2 different structure difference. Now, if the major point here is structure, the word is structure between the monomer, and they are saying about the monomer of DNA and RNA. So here they are talking about the monomer, and if you look at the monomer, they are looking at the nucleotide of DNA and the nucleotide of RNA. So you only look at the structure which is the nucleotide, of which the nucleotide is made up of a phosphate, a sugar, and then the nitrogenous base. So there are only two differences here you can look at. You can look at the sugar. DNA nucleotide has deoxyribose, deoxyribose sugar, while RNA has ribose sugar. And then DNA has thymine, while RNA has uracil. So after that, you draw a table. It must be a complete table. Yes, it must be a complete table. And then you put DNA, and then you put RNA at least. Then you get a tick here, a tick here, a tick here, a tick here, and a tick for the table, which makes it to be 5 marks. And if you find a question like this and tablet, and there is only 10 minutes or 10 seconds remaining, don't waste time. At least you can grab that 1 mark. Just draw a table like this, and then put DNA, and then put RNA. Yeah, even if you don't have anything here, then we shall come and give you a tick. At least you score that 1 tick. So yeah, please try to use different ways in which you can get marks. Yes. Yeah. So this is the same thing we have been trying to discuss. Phosphate, you have seen them, and then M2 process, which require the strand of the nucleotides in a single strand of DNA. Sometimes people think that DNA can be a single strand. It can be a single strand. What makes this one to be DNA is because of the thymine, which is being indicated there. So thymine is in this form, while adenine is in that form. Then they're saying that how many nucleotides are represented in the diagram? Yes, let's try to answer this. How many nucleotides? A nucleotide is a phosphate, nitrogenous base, and a sugar. So this is one nucleotide, the second nucleotide, the third nucleotide, and then this is the fourth nucleotide. So the answer is going to be 4 nucleotides. And then they're saying phosphate molecule, then phosphate molecules, you just count them 1, 2, 3, 4. So the answer is also 4. And then they're saying that how many nucleotides, okay, I done that. Sugar molecule, there are 4, but sugar, this is the phosphate, and then this is the sugar. Write down the nitrogenous base from top to bottom as indicated by the arrow as complementary base strand and this molecule. Here is just T, they're saying on DNA. Complementary. So they are not talking about convert it into RNA. Don't forget that A goes with the T, then C goes with G. So whenever there is A, T, we put A. Whenever there is C, we put G. Whenever there is A, you put T. Whenever there is C, you put G. So basically, that's how it's supposed to be answered. Name 2 processes that require the DNA strands of DNA molecule to separate into single strand as shown in the diagram. Which processes? 2 processes. We only find this in when the DNA replication, DNA replication is going to take place, and also we find this when there is a transcription. Remember here you form 2 strands, 2 separate strands. Each strand is acting as a template. While here you form 2 separate strands. Only one strand will act as a template. And while here 2 strands will act as a template. So these are the 2 processes which make DNA molecule to open. DNA profiling is also not a big deal. Technique, you have to know how to identify this. Read the question very well and then find out. Don't just answer the question. Detectives were investigating crime scene, found the blood on the broken window. Then they suspect that the blood was that of the criminal. To identify the criminal, they analyzed the DNA sample from different DNA sample from the blood and compare it to the 4 suspects. Now, look, this is DNA sample, and then these are the suspects. It means that these are the DNA they found on there, but you have to see where it is corresponding. This one goes with that. This one goes with that. Still, yeah. This one is that. Use a ruler to identify this. That this with this. This with this. This with this. This with this. So it means that Jenny is a suspect for that case, whose DNA profile is matching with the DNA sample obtained from the crime scene. Name the technique which was used. It's DNA profiling technique is DNA profiling, and what is being shown here is a DNA profile. So you must know the difference. DNA profile is not a technique. DNA profile is what you obtain when you analyze the DNA sample. So who was the possible criminal? We have seen it is Jenny, because the DNA profile, or the DNA profile of bars there of sample, DNA sample profile is matching with the profile of Jenny, or the band of DNA profile sample, they are matching with that of Jenny. So state one other use of DNA technique, or state other use of the technique identified in the question above, meaning that the DNA profiling, what is it used? Can be used to identify the relatives. They have talked about the criminalism, so you don't talk about that anymore. So talk about the identification of the perfect organ donor. You can talk about lost relatives. You can talk about diagnosis of the inherited diseases. So there are so many uses you can use to when you are dealing with the DNA profiling. So it's the same thing. You see that is the same thing. I don't usually take too much time here because these questions are really open. Who is a suspect? You know how we have identified it. What diagram is DNA profile? What does the diagrams above represent? They're not saying the technique, they're asking for the diagram. Therefore, it is DNA profile, not DNA profiling, because here they said which technique, name the technique. So it's DNA profiling, but here it is DNA profile. Which suspect? Look at the bands and then you match them, give a reason. So the suspect, if you look at them, you'll find out that Q is most likely to be the suspect because the bands of broken glass, they are matching with that of Q. So they're saying that name 2 possible disadvantages of using this evidence in the court. It might not be accurate 100% because number one, they can human error, number one, and also it may not be unique if a small sample is used. Also, they can a human being can manipulate them, so it may not be 100% accurate. So we have seen many questions concerning about DNA profiling. Download this and then try to do these questions. Where you get stuck, come back to me. Ask me questions. I'll be able to help you out. My number is always on the website 0848331738. Yes, and then speak to M.Saidi, who is always ready to help you out. Then another question is still the same tablet. Whenever you talk about tablet, make sure that you draw a nice table and then so that you get that 1 mark. That's why there are 5, because they're asking for 2. They give you 2 marks there and then 1 mark for tabulating. Describe the process that is being represented or responsible for formation of molecule B. So here, molecule B is messenger RNA. Why messenger RNA? It's because of this. So this is not, you see, this is messenger RNA, which is uracil. Therefore, and also 1 strand is being used. So that's evidence to show that this is transcription, because they are saying this is non-coding strand. So it means that only this is acting as a template. Yes. So they are saying that describe, so what are they looking for here? Describe the process that is responsible for formation of B. If you look at B, is messenger. So you describe. How do you describe here? Let me just talk about it as I told you. Talk about DNA double helix, unwind, weak hydrogen bond, break, and unzip. So when the weak hydrogen bond break, it's the same thing saying unzip. So you get 1 mark there. To form 2 separate, the word separate must be there, 2 separate strands. Only 1, you see it here, that only 1 is acting as a what? Is acting as a template. Only 1 is acting as a template. So it's being shown there that this one is acting as a template, and then using free floating messenger RNA. So you have to write messenger RNA nucleotide. Yes, to form a complementary strand where adenine goes with uracil and then guanine goes with cytosine. Adenine goes with uracil and then guanine goes with cytosine. The complementary strand move out of the nucleus via the nucleopore to the cytoplasm on the ribosome. So basically, that's what you're supposed to explain. Please don't try to bridge it, because it's a process. One step leads to the other. If you start with what is on at the bottom and then you start with it, then you lose marks because the sequence is not the same. So you cannot start with the last step and then you end with the first step. You have to start with the first step and then you end with the yes. So molecule B, we said it is messenger RNA. Name the process is transcription. Explain how the protein molecule would be affected if codon GUU is changed to CGC. So if here it depends on two things. If there is a table, yes, now you compare the table and see if it's gonna lead to a different amino acid. But because now this is open, then we assume that it's gonna be a different amino acid. For example, if you say that you started with GUU, yes, and then GUC, from this to that. If this, let me explain this. If this is tryptophan, yes, codes for tryptophan, and this one also codes for tryptophan. However much this has been changed to this, is going to code for the same amino acid. Therefore, the functioning of the protein is not going to change. But if this tryptophan and then now this one codes for example, leucine, then it means that there will be a change in the functioning of the protein, because this is tryptophan and then this is leucine. Therefore, a different protein will be changed, will be formed, and then still the functioning of the protein will also change. So explain how the protein molecule would be affected if this is changed to this. So therefore, you tell us that if GUC is changed to GUC, GUU is changed to GUC, because they say that codon this changed to, because this, when they say codon, it means that we have started from the messenger RNA. Then this will lead to different sequence of amino acid. Different amino acid will be brought to the molecule or to the messenger RNA, not messenger RNA, will be brought to different amino acids will be brought to ribosome, and then a different protein will be formed. So that's basically you have to understand. And then when you come here, this is translation, because it is occurring on the ribosome. So what is A? A is messenger RNA. What is B is a ribosome. And then what is C? C is a transfer, RNA. What is Z? Z is the amino acid. So this is the monomers, amino acids, monomers of the protein. And then this is the transfer RNA. You have seen it. And then this is the ribosome, and then this is the messenger RNA. So basically, you need to do that. And then what is this? These are 3 bases. So therefore, these are codons. Yeah, remember, the 3 bases on the DNA, we call them cods. And then 3 bases on the messenger, we call them codons. And then 3 bases on the transfer, we call them anticodons. Organelle X, what is organelle X? We have seen it. We have seen all the questions. You see now, when you start with trying to label them, you'll be able to answer all the questions. Molecule W formed, you see, organelle Z, X, we have seen it, ribosome, monomer W, the monomer. They're saying the monomer of molecule W. Remember the building blocks, that's what we call the monomer. Therefore, it's going to be the nucleic, the nucleotide. Be the nucleotide. Yes. And then molecule W and Y, W and Y, we have seen it. W is messenger RNA, and then Y is the transfer RNA, and then molecule or organelle X, we have seen it is a ribosome. So still the same thing. You can see Some people, they call this a protein. This is not a protein. It is only a protein if it is 50 and above. And above amino acids. So what is this then? This is a polypeptide chain. Yes, so it is a polypeptide chain. So what is X? This is the amino acid. And then what is this? This is transfer RNA. And then what is Z? We have seen them. These are anticodons or anticodon. And then this is messenger RNA, and the 3 bases on the messenger RNA, we call them codons. You see, you can't fail such questions. When you come to the question, you see that they're asking you the exactly same thing, and then asking you describe the process of transcription, which we have described. So basically, at least you have some light on DNA, and then maybe we will talk about the process of translation, and then after that we wind up with this coding and then the mutation. These are some of the short question which you need to know before you go for the exam. Number 1, DNA replication. You have to know DNA replication, the process by which DNA makes the exact copy of itself. Where does it occur? We have seen it in the nucleus. When does it occur? We say that it occurs during interphase. And then we said, why does it occur? We say that to double the chromosome number. And then how does it occur? That's the process of DNA replication. And don't forget that here you have to explain that here the tools, you form 2 separate strands, and each strand is acting as a template, and that is the major difference between DNA replication and transcription. So in most cases, people confuse transcription and the DNA replication. Then number 2, describe the location and the structure of DNA. Here I explained it at the beginning, the structure of DNA. In most cases, give it 6 marks. And then location, we have 3 locations, that is nuclear DNA, mitochondrial DNA, and chloroplastic DNA. So if they talk about in plants, then you talk about all of the 3. But if they talk about in animals, you only talk about the 2, that is the because in we don't have a chloroplast in animals. So describe the structure of RNA. Structure of RNA, it's a little bit the same as DNA, but only few things which are not there. That is the thymine, the double strand, and then also the weak hydrogen bonds. Describe the involvement of different types of messenger RNA process in protein synthesis. Here they are talking about the functions of each. For example, you talk about transcription and then translation. So it is like you are describing the whole process of DNA of protein synthesis. Another thing you need to know here is also describe transcription in detail, and then also translation also in, yes. And then also what they can ask you is why is the mutations which occur. And I say that if you change the code, for example, from GUA to GCA, then if these two they code for the same amino acid, therefore the protein will not change. The structure of the protein will not change. But if the different amino acids are being coded for, then the structure of the protein will be changed. So let's go to meiosis. Let's go to meiosis. There are so many questions which can talk about DNA. And also the base calculation, whereby you're supposed to talk about if adenine is equal to 10%, what is the percentage of guanine, or the ratio of guanine, whereby you have to tell us that adenine goes with the thymine, and then cytosine goes with the guanine, whereby if this is 10, or definitely this is 10, and then 10 plus 10 is equal to 20. Since this is percentage, therefore I'm going to say 100 minus 20, which means 80%. And then 80%. So what happens? So it's going to give us 80% for these two. So divide by 2, and then from 40%, therefore this is 40, then this is 40. So this is just a simple recall. I hope you remember. Please don't go without knowing it, because at least those 2 marks you'll be able to answer them. And then if they are looking for the ratio, you can put them maybe adenine now. Adenine, we said it is 10, and then maybe with the guanine we said is 40. So divide by 10, and then we're going to get 1, 2, 4. So sometimes they swap it and they say 4 to 1, and then because when now you have seen 1, and then you have seen 4, you have seen 4, you have seen 1, you just circle that. It is what? It is wrong. So when you go to meiosis, so many point where crossing over takes place. We call it chiasma. You see, a chiasma. Splitting of the cytoplasm, we call it cytokinesis. Okay, we can answer them. Failure of the chromosome to separate during meiosis, the non-disjunction, the structure in the cell that forms spindle fiber, centrioles, and then the phase of meiosis where homologous chromosomes are aligned along the equator, that is metaphase 1, because they're saying homologous chromosomes. Then the point at which the 2 chromatids of the chromosome join, we call it centromere, and this division of the nucleus. Division of the nucleus is karyokinesis. And then they're saying that the site of meiosis in a female, because in female we find meiosis in ovary. So you write ovary there. Ovary. And then the exchange of genetic material between chromatids in the homologous chromosome, which is crossing over. And then they are saying the genetic disorder caused by extra chromosome on chromosome number 21, that is Down's syndrome. Yes. And then they are saying that the structure formed between the centrioles by the centrioles during the cell division with spindle fibers. Yeah, spindle fibers. And then they're saying that non-sex chromosomes in human, we call them non-sex autosomes. And then they're saying the condition in the cell where there is only one set of chromosome. How do you call that condition? We call that condition haploid. And then the structure that is responsible for formation of the spindle fiber during spindle fiber, we say that is called the centrioles. Yes, and then the A chromosome aligned along the equator, none, because here it must be metaphase. Random arrangement of chromosomes, it must be metaphase. So it's A, B only. The site of meiosis in plants, anthers, and ovary, both in, this is male, this is female. And then the process of meiosis produces meiosis result in 4 haploid daughter cells, 4 diploid. So this one becomes wrong. So this is the correct one. The mechanism to introduce genetic variation, crossing over, is correct. Random arrangement of chromosome is also correct. So it is both A and B. So basically, you see, yeah, so now we are starting the other part of meiosis. All right. Please don't forget to subscribe, stay in touch. If you have any problem, we need to smash these questions, and then we get all the marks at the end of this period. Yeah, when you come to meiosis, there are some questions we can talk about. Let's see what we can go through so that they can also benefit you in answering questions related to meiosis. I know it is a simple topic, but is a little bit challenging to some other. But let's see if we can smash these questions and then we get a better understanding. If you don't understand the topic, let me know. I'll help you out so that at least we can have more distinctions in this year. All right. They are saying that here you are seeing diagrams. What comes into your mind is the kind of phases which this diagram is indicating. So, what is C? C is the spindle fibers. What is B? This because there are two chromosomes of the same kind or the same size and structure, therefore is homologous chromosome. Then the joint between two, or the joint between two chromatids of the same chromosome, it is a centromere. And then diagram A, what does diagram A represent? Because of this one is starting to form, so it can represent prophase, prophase 2. Why prophase 2? Why not prophase 1? It's because now this, they are individual, they are not in pairs. And then number 2, you see that crossing over took place a long time ago. And then this one is metaphase. The chromosomes are aligned. If you turn this, and then comes this side, and then this goes that side, so that this thing is in between your chest. So you see that if this is one side of the chest, and then this is on the other side of the chest, I think now you see what I'm trying to say. So you see that they're in the middle. So why? These are the spindle fibers. Yes, if this is the spindle fiber, if this is the spindle fiber and this is the spindle, sorry, centrio. So these ones are in the middle. That's why you say that it is under metaphase. But metaphase, which metaphase? It is metaphase 1 because these chromosomes are in the middle. Then they say identify A. We have seen it. A is centromere. B, what is B? B, we have seen it homologous chromosomes. Then C, you see, we have seen it as a spindle fibers. Identify the phases in diagram 3. Diagram 3, because they are separating. Yes. So if they are separating, then they are moving to opposite poles. You see them? They are moving to opposite poles. Therefore, it is anaphase. But which is it? Anaphase 1 or it is anaphase 2? Is it anaphase 1 or anaphase 2? Because now there are chromatids which are moving to opposite poles. Chromatids moving to opposite poles. Therefore, it's going to be anaphase 2. Anaphase 2. And then they're saying that if it was anaphase 1, then this would have been individual chromosome. This would have been a chromosome moving to the other side. Also, this one would have been a chromosome, and the centromere there, this would have been a holochromosome, and also this would have been a chromosome. So it would have been anaphase 1. But because their chromatids moving to opposite poles, therefore it is anaphase 2. Then they're saying that write down the number of the diagram shown in the sequence which the phase occurs. The number of the sequence. They're saying write down the numbers of the diagram to show the sequence in which. So basically, we have to look for meiosis 1. The chromosomes are supposed to be in homologous pair. So this one becomes, this is homologous pair. So it is 1. Therefore, it is what? It is metaphase 1. And then this one is prophase 2. And then after prophase 2, they separate, which is, so it's going to be 1, it's going to be 2, and then it's going to be 3. So when you are writing the numbers here, it's going to be diagram 2, diagram 1, and then diagram 3. So it's going to be diagram 2, diagram 1, and then diagram 3. Then one difference between Metaphase 1 and Metaphase 2. In Metaphase 1, the chromosomes are aligned along the equator in homologous pairs, while in Metaphase 2, chromosomes are aligned along the equator singly. They are individual chromosomes are the one which align along the equator. So that is the major difference. And then if we see another question, so label it. Very simple. You see, we usually bring the same kind of questions. So this is spindle fibers. This is centromere. This is chromosome. Non-homologous chromosome is one chromosome. And then this is centriole, which is being produced by centrosome. Okay, centriole. And then what causes these differences in the chromosomes or chromatids is because of crossing over. When does crossing over takes place? It takes place during prophase 1. How does it take place? Homologous chromosomes, they align together. Chromatids of the homologous chromosome, they cross over at a point called chiasma, and then they exchange the genetic material, and then they separate to form different chromosomes which are genetically different. Basically, that is the process of crossing over. You can't go to the exam without knowing the process of crossing over. Identify the phase of meiosis in diagram 1. Diagram 1, because now, yes, the chromatids are separating, is the same thing like this one. It's the same thing. The same thing like this is exactly the same. Yes. So it's gonna be Anaphase 2. And then this one is going to be Metaphase 2 because the chromosomes are aligned along the equator singly. Yes, they align along the equator singly. Then they're saying that identify the phase. You are done with that. State what happens to structure D in the next phase. Where is structure D? Structure D, these are the chromosomes. So the chromatids, they move to opposite pole due to contraction of the spindle fiber or due to the shortening of the spindle fiber. Name the process during which genetic material were exchanged. What was exchanged? We say that the process is called crossing over. And then we said it crossing over. And then we said it occurs during prophase 1. State the consequence if the mentioned does not occur. If this process does not occur, it means that organisms, it will reduce what you call the genetic variation. It means that more organisms will be more similar to each other. So it will reduce variation among the organisms because crossing over leads to genetic variation. Then they're saying that give the number of the chromosome present in the original parent cell in this diagram. They didn't say this is a human cell. I don't see anywhere there saying this is a human cell. So you have to know, you have to use the number which is being given. So because this is meiosis 2, and meiosis 2 leads to the diploid state being halved. So for example, you start with 1N, and then you form 2 cells, which is 1N, and then another 2 cells you divide to form 1N, 1N. So if you start with the 4 here, therefore it's gonna be 2, it's gonna be 2, it's gonna be 2, it's gonna be 2, it's gonna be 2. So it means that this is meiosis. Therefore, this is 1, this is 2. Therefore, the total number is 2. Please don't talk about 46 if they didn't say that this is the human cell. And then now they are saying original cell. So if this original cell, therefore it's gonna be 4. And then you see now they are saying specifying it. Human cell in the sample diagram 2. Because diagram 2 is haploid, therefore it's gonna be 20. It's gonna be 23 chromosomes. So basically, that's how you're supposed to answer such questions. So you can answer these questions, at least in meiosis. What is A? A, in most cases, students confuse it with a nuclear membrane. This is not a nuclear membrane. You can't have a nuclear membrane when the cell is under a metaphase. Therefore, this is the cell membrane. This is the chromatid. This is the chromatid. This is the spindle fiber, and then this is the centriole. So this is metaphase, metaphase 1, while this one is anaphase 1. Why? Because chromosomes are separating. What about this? This is the telophase, telophase 1. Why? Because they are chromosomes. They are chromosomes which are still double-stranded. Yes. Or at the beginning of prophase 2. Yes. So you see that now once you name that, you are able to answer this question. What is phase 1? We have seen it is metaphase 1. What is 3? We have seen it is telophase 1. Then you're saying the letter only of the part contains DNA, contains DNA. Give the letter of only part that contains DNA. It is B, because in this case they didn't mention any chromosome here. It's only B, which is chromatid, and then attaches the centromere of the chromosome, attaches the centromere chromosome, which is C, attaches is the centromere. Yes, and then they're saying that forms spindle fiber, which is the centrioles. Where is the centriole mentioned? Is D. You see, we are able to answer these questions. Name the organs in humans, male, where meiosis occur. In humans, male, it occurs in the testes. While in female, it occurs in ovary. In plants, it occurs in anthers, that is male, and then stigma, ovary in female. So here they produce sperms, they produce ovum or ova. Here they produce pollen grain, while here they produce ovules. Yes. So basically that's a simple illustration of meiosis occurring in plants and also in animals. Describe how meiosis leads to genetic variation. So now here you are talking about only processes which talk about genetic variation. Here you talk about crossing over. So you describe crossing over. Then you talk about the random arrangement of chromosomes during metaphase. Metaphase. Yes, so basically that's what you need to explain. It can lead to genetic variation. You can also mutation can also be part of meiosis. Why? Because if you talk about Down syndrome, it occurs during the process of what? Occurs in process of meiosis. But these two, they can help you to get those marks you are looking for. Here is an example of a karyotype. You have two individuals. Identify which karyotype. Here, when you're given a karyotype, look at, is there any abnormality? Which one is the male and which one is the female? So if you look at this, no abnormality. All of them, they are in pairs. But if you look at that, one is long and then one is short. Therefore, this is X. This is X, and then this is Y. So therefore, this is a male. And then both of them are the same size. Therefore, this is a female. So now from there, you can now go and then try to answer. What term is given the chromosome number 1 to 22? So we call them, because they can ask you to describe the structure of a karyotype. You call them orosome. The orosomes. And then 23, these are gonosomes. Why? These are sex chromosomes. Yes. State the gender of P. We have seen it. P. Where is P? Male, because of this. Give one observable reason, because the chromosome number, the chromosome number the chromosome pair number 23. One chromosome is bigger than the other. Therefore, one is X, one is Y. Hence, the person is a male. Then they're saying, give one observable. We have seen it. Each of the pairs shown is a homologous pair of chromosome. State the origin. State the origin of each chromosome in the homologous pair of the chromosome. So here, where do they are two. Where are they coming from? These pairs, one is coming from the father, and then one is coming from the mother. Yeah, that's why there are two. That's why we say that when the sperm comes, is haploid, and then fuses the ovum, which is haploid. So one chromosome, another chromosome, when they combine, you form the pair. So it's the same thing. Sperm, ovum, sperm, ovum, sperm, ovum, sperm, ovum, like that, like that, like that. So one comes from the father, one comes from the mother, and then form a diploid, a zygote. Then they are saying that the diagram below represents the distribution of chromosome 21 as it appears in the gamete. So it's the same thing, guys. Yeah, so you see that you always find almost the same diagrams, so same structures. So now draw a diagram to show the cell during Anaphase 2 of meiosis. Now here, when I ask you to draw a diagram, you must write a title. That is the first thing, title. You cannot draw anything without a title. Use a sharp pencil. It must be neat. Yes. Someone is going to ask you, am I neat when I'm writing? Yeah, I'm saying you must be neat. I'm not a student. All right. I'm saying that. So when you draw, they're saying this is the Prophase 2. Prophase 2, why? Because they are single. So what do? You draw the cell, and then they're saying during anaphase. So it means that before it's going to be like this, and then the short one down. So this is prophase. Just drawing a structure, but you have to show the variations when I'm drawing. So during anaphase, it means that one, and then one has a variation. So it's still moving to the poles, and then also then the short one is gonna be the short one, and then it's also moving, you see, but it also has the biggest variation, and then this other one is completely shaded. You see, you have to show that. And then this is spindle, this is chromatid moving, and then and then this is the cell membrane. Then write the title. Yeah, at least you are able to answer such question. Yes, so that when you come, we'll maybe give you a mark for title. Yes, a mark for drawing, correct drawing, and then maybe those are 2 marks. Now, maybe 3 marks for labeling. Yes, so that's why I'm saying labeled diagram. So meiosis is also easy. If you look at most of the questions, you will see that we have answered those questions. Can you answer this? Yes. 10 seconds, 8 seconds, 7 seconds, 5 seconds, 3 seconds, 1 second. Okay, let's see. Then they're saying that this is what? Spindle fibers. What is this? Homologous chromosome. What is this? Centromere. Yes. So these are homologous chromosomes. And then this is the centriole. And then this is the cell membrane. So you can answer these questions. You see, nothing which is going to be new in your paper. Name the process diagram above. You see, it is metaphase. You see, you have to turn it so that these things are in the sides. Check. You see, they're in the sides. So that you see now clearly it is metaphase. Name the process, name the phase, represent a diagram. We have seen it. Then they are saying name part C. We have seen it also. Centromere. Then B represent homologous chromosomes. B represent what is homologous chromosome? Chromosomes which have the same structure and the same size, and then they carry genes. Genes of the same characteristics are always on the same locus. Yeah, so we call them homologous chromosome. Then they are not genetically identical, but genes of the same characteristic, they are in the same locus. Locus is a position of a gene. It's singular. Loci when they are many. Explain the appearance of chromosomes in diagram. Why are they shaded like this? Is due to crossing over, which took place in prophase 1, whereby the chromatid exchange the genetic material. That's why they are like that. They are saying that total number of DNA is the total amount of DNA is 12 viral units each daughter cell at the end of this cell division. How many DNA was in the parent cell? So they are saying that the total number of DNA is 12 units in each daughter cell. Remember, you start with the 2n. You start with what? 2n. Yes. So 2n, you form two cells, 1n, then you form another two cells, you form another two cells. So 1n. So if you started with the, if your daughter cells are having 12 here, so it means that then if you want to get the parent, is times 2, so which is 24. So the answer is going to be 24 DNA binary units. Don't forget to put the units as they are being given to you. So many questions, you'll find them like this. This is the still a karyotype. Male or female is a female, while chromosome number 23 is having chromosomes which are of the same size and the shape. See? So describe the karyotype of person with Down syndrome. So a karyotype with Down syndrome will have an extra chromosome on chromosome number 23. Yes, so the pair chromosome, so there will be 3 instead of what? Instead of 2. So you see that these diagrams, these questions, you can answer them very well. Yes, you can answer them very, very well. All right, let's just do few questions about it. What is this? What is this? This is a centromere. What is B? Is a crossing over point where crossing over takes place. Therefore, is chiasma. These two chromosomes under crossing over, we call it a bivalent. Bivalent or homologous chromosomes during crossing over. Then this, what is this? This is a chromatid, and then what is this? Is a chromosome. So these are sister chromosomes. So now you can answer the question. Describe the process that occurred at D. So it means that they're asking to describe the process of crossing over. I explained it before. This is where tricky things come from. They're saying the certain number species has 42 chromosomes. They're not saying it's human. So don't talk about 46. Yes, please don't talk about 46. They are saying 42. The muscle cell, its muscle cell, remember muscle cells, those are autosomes. They're always 2n. Yes. Give the chromosome number of each of the sperm cell. So it's going to be half because sperm cell is 1n. Therefore, the answer here is going to be 21. The skin is autosomes is 2n. Therefore, it's going to be 42. And then ovum of the female of the counterpart is the same thing. Is going to be 1n, which is 21. In most cases, the number of chromosomes are the one which are confusing students, and they end up losing marks. So it's the same thing here. Crossing over took place. They can ask you to draw the diagram after crossing over. So members, students, please don't lose marks. Don't lose hope. You will make it. You will make it. I'm going to come back for genetics and inheritance. And then after that, I will go to human evolution. Thank you very much for watching. Don't forget to subscribe, like, and share. Don't also stop to comment so that I know where exactly I should put more effort and where to help so that you can get your distinction.